Okay. Good morning, everyone. Good afternoon, and welcome to the Global Nurse Family Partnership Webinar Series. My name is Susan Jack, and I'm a professor at the School of Nursing at McMaster University in Canada, as well as I am the Global Nurse Family Partnership Research and Evaluation Director. And it is truly my pleasure to welcome all of you from around the globe to join us today for this exciting webinar. And as we begin this virtual meeting, I would like to take a moment to acknowledge that I am joining all of you today from an area that is called Guelph, Ontario, in Canada, which is situated on the treaty lands and the territory of the Mississaugas of the Credit First Nation, and which is part of the Dish with One Spoon Wampum Agreement, which is a commitment to share and care for this land in peace. And this acknowledgement reminds us of the enduring presence of Indigenous peoples on this lands and their contributions, both historical and contemporary. As this is an international and global gathering, I also encourage each of you to reflect on the lands that we all occupy and the Indigenous people who have cared for these places since time immemorial. Together, let us commit to learning about and advancing reconciliation, equity, and justice in all of our respective contexts and lands. It is my pleasure now to introduce Dr. David Olds. To many of you, he will need no introduction. And for those of you who are new to the Nurse Family Partnership community, Dr. Olds is a professor in the Department of Pediatrics. He was the founder of the Prevention Research Center at the University of Colorado, Denver. Dr. Olds is globally known and renowned for developing the Nurse Family Partnership Program, even before it was called the Nurse Family Partnership Program, and leading evaluations of this program for over 40 years to develop the evidentiary foundations upon which it rests. And David has been an inspirational leader to governments, to policymakers in many countries around the world who have made the decisions to adopt and adapt and implement and continue evaluating NFP in global contexts. And he has been an inspiration to many scholars, including myself and our guest speaker today, Dr. Andrea Gonzalez. So David, I turn it over to you to introduce our speaker today, and over to you, David.
Sue, thanks so much for that introduction. It's my pleasure today to introduce Dr. Andrea Gonzalez to all of you. Andrea is an associate professor in the Department of Psychiatry and Behavioral Neurosciences at McMaster University and a core member of the Alfred Center for Child Studies there. She holds a Tier 2 Canadian Research Chair in Family Health and Preventive Interventions. And currently, Dr. Gonzalez is a member of the Public Health Agency of Canada's Child Maltreatment Surveillance Working Group, as well as a member of the Board on Child, Youth, and Families for the National Academies. Her research focuses on the developmental consequences of early adversity, intergenerational transmission, and evaluating evidence-based preventive interventions. She's involved in several evaluations assessing the effectiveness of preventive interventions with young families, where she collects both behavioral and biological measures from caregivers and their children from birth through age 6. Andrea's presentation today is from the Healthy Foundations work that grows out of the British Columbia Healthy Foundations or Connections randomized trial. And as you'll see, Andrea is a wonderful, thoughtful person. So, Andrea, take it away.
Great, thank you so much. I'm just going to get my slides ready here. And while I'm doing that, I'd just like to thank Susan and David for the invitation to come and present to you today some of our preliminary findings, as well as Anna for all her amazing organizational efforts. And I know I was supposed to present in September, and I apologize. I got COVID, so we had to postpone. But I'm really excited to be here today. So I'm just going to check, Anna, if you could give me a thumbs up that you could see the slides. And okay, awesome. So thanks so much. Today I'm going to be presenting, as David mentioned, on the Healthy Foundation study, which was an adjunct study that was built into the British Columbia Healthy Connections project, which I'll briefly touch on it as well, which is a randomized control trial that was done in the province of BC here in Canada. But I'm just one person presenting, but backed by an amazing large team. And the BCHCP, which was the randomized control trial, was led by Charlotte Waddell and Nicole Catherine in British Columbia. And the co-principal investigators were Harriet McMillan and Susan Jack and Debbie Sheehan, as well as a number of other investigators. From the Healthy Foundations team, there are a number of other investigators, and I'd like to particularly point out Dr. Divya Joshi, who's actually attending today, as she really has dug deep into the data and the analyses and has helped me think about this all very, very thoughtfully. And then I'd like to really thank all the NFP public health nurses and supervisors in BC, the health authorities, the steering committee, and the data safety and monitoring committee, the provincial advisory committee, and of course all the staff from both studies and team members. And finally, all the mothers and children that participated in this work and trusted us with their information and even the biological data that I'll be presenting today, which could be quite anxiety provoking for these new moms. Both of the studies received funding, which I've listed here. The Healthy Foundation's funding came from the Canadian Institute for Health Research, and a lot of the funding for the BCHCP came from the BC government, as well as the health authorities that supported the project as well in British Columbia. So my main objectives today are really to provide a broad framework. Why would we even look at biological markers in moms and their infants within the context of a preventative intervention like the Nurse-Family Partnership? So it's to give you a step back and a broad overview of the field at large, leading down to the rationale for the Healthy Foundations project. Then I'm going to talk a little bit about the study design and the measures that we collected, as well as the main preliminary findings. And I'm hoping that we could have a really fulsome discussion near the end. I'm hoping I left enough time so that I could get your feedback about some of our preliminary findings, and also I know there are a lot of providers and practitioners who are on the call today, what this means in the context of your own work and what else you might want us to look at in terms of the data. So we know that the building blocks for both for healthy development are established very, very early, and both animal and human research tells us that early nurturing and protective relations are essential for our basic biology. So that early nurturing and those responsive, sensitive interactions that caregivers have with their infants really provide the emotional security and support that children need to explore their environments and learn, but also those interactions help stimulate the brain and the neural connections that are underlying the brain. And we know that genes are the main driver behind initial brain development, but genes don't design the brain completely. So the functioning of the brain actually gets fine-tuned through environmental influences. So that could be broad influences like exposure to different kinds of pollutants or chemicals, both in utero and postnatally, but also really importantly, it involves the interaction, with the caregivers and the environment as well. And we know that in the first year, there's incredibly rapid brain development that's going on. So in the first year, the brain actually doubles in size, and that this occurs through a proliferation period where 700 new neural connections are made every second in the first few years of life. Brain development or learning is this process of creating and strengthening these connections between neurons. And these connections then form the basic kind of structure of the brain, the circuits and connections that underline all the learning. And our experiences and the environment that infants are growing up in sort of dictate which circuits and connections get more use. So connections which are used more grow stronger and more permanent, while connections that are used less tend to fade away through a process called pruning. So the pruning process is when connections are reduced so that the brain becomes more efficient. We can't have all of these connections in the brain all the time. So the environment is really shaping the way the brain is structured through both the proliferation period, but the pruning period as well. And sometimes we refer to this as the concept of use it or lose it. So that if you're not making those kinds of connections, then the pruning sort of goes away and those connections are lost. By age 3, about 85% of the brain has reached its adult size volume. And even though the brain remains plastic for much longer than we originally believed, it's much easier for the brain to respond to external environmental stimuli and more effectively very early on. And while this proliferation and pruning process is underway. So we know that accompanying these, very rapid brain changes are changes also in learning and development in the child. And this kind of goes according to a prescribed process where sensory pathways come online first, very early on within the first few months, followed by early language skills, and then higher cognitive functioning, which actually we now know goes into our mid-20s. And it's these simpler circuits build, more complex circuits sort of build upon these early simpler circuits. So again, even though the brain maintains like plasticity over time, it does become more and more specialized and more difficult to adapt and reorganized over time. And we see this through things like language, for example. It's very easy to capture language and to learn language in our mother tongue, in the language that we hear as infants. And it's much harder for us to learn language later on. So it's not impossible to learn a new language when you're older, but it becomes more challenging. So the question is, what happens when this foundation is at risk or if it's compromised? So what happens when things might go wrong or those fundamental early relationships are not established, or if there's a high stress situation that's created for the developing infant? We know that caregivers are a key regulator of infant stress and that they can be a buffer between other environmental or external stressors and the infant. So I really love this taxonomy that was developed by Jack Shonkoff at the Harvard Center for the Developing Child, because he really talks about how not all stress is equal. Like when we think about stress, we typically think it's a negative thing, but really we need stress in our lives. And that's what positive stress is. Positive stress are those normal, typical childhood experiences that kids need to experience in order to develop some kind of resilience. And in these kinds of cases, like being dropped off at a daycare or your first day of school or losing a game, there isn't any buffering that's needed from a caregiver, and it really involves just temporary, mild, increases in our stress levels. But it's helpful to have these kinds of stressors because it helps us develop coping skills and resilience. Then there's tolerable stress, which might be more complicated and scary. So maybe exposure to a natural disaster or a death of a loved one that's not necessarily a parent, or financial strain, not poverty and chronic poverty, but financial strain. And a caregiver can buffer the stress of the child. So this might be more continuous kinds of stress, but the caregiver helps regulate the child's stressors, and there's a good chance that the child will recover and their underlying biological system won't be impacted. What we really worry about is what's called toxic stress, and these are the severe, sort of long-lasting, chronic types of stressors that a child does not have any kind of control over. So it's things like adverse childhood experiences, so exposure to child maltreatment or to intimate partner violence or severe economic hardship. In many cases, there's not an adult that is available to buffer the child's stress, and this leads to prolonged activation of the stress system, which has a trickle-down effect, which I'll talk about later. And we know that these toxic stressors or adverse childhood experiences really increase the risk of negative outcomes across the lifespan. So early in life, we might see an increased risk of injury, attachment difficulties, or developmental delays. In early and middle childhood, we might see externalizing behavior or internalizing, so difficulties with anxiety and mood, as well as some physical symptoms like asthma. And then later in life, there's a perpetuation or increased risk of mental health difficulties or physical health difficulties, and difficulties potentially in parenting one's own child. So if we take a life span approach and we sort of put it on its side in this model of this pyramid that comes from the Center for Disease Control, it's really argued that these adverse childhood experiences early in life sort of impact social and emotional cognitive impairment, which increases the risk of adopting health risk-taking behaviors like smoking and drinking, which leads to increased risk of disease and disability and potential early death. But this model does recognize that there are scientific gaps in this pyramid, and really those scientific gaps are about biology. So this is, again, a diagram of a framework that comes from Jack Shonkoff from the Harvard Center for the Developing Child, where they really argue that both positive and negative experiences sort of interact with genes to lead to physiological adaptations and disruptions, which then are relate to later outcomes, health behavior outcomes, or education, or mental health outcomes over time. And they focus on two specific pathways in which early experience sort of get built into the body or get under the skin, like Clyde Hertzman used to say. One pathway is through cumulative effects over time. So this is the wear and tear on biological systems that's caused by recurring adversity. And this is specifically related to chronic activation of the body's stress system. So our body has two main stress systems that are activated. One is the sympathetic system, which is faster acting and results in the release of norepinephrine. So it's like when our heart rate starts to increase immediately when we're stressed or we start to sweat. And the second pathway is the hypothalamic pituitary adrenal axis, and that's the slower acting pathway. And it essentially sets off a cascade of hormones that eventually lead to the release of cortisol at the end here from our adrenal glands. And when cortisol is released to the body, it essentially galvanizes the body to kind of act and deal with the stressor. So once cortisol has been released and the stressor has been dealt with, there's a negative feedback loop, which is this red arrow here, that goes to the other systems within the HPA axis and says, okay, the stressor's gone. You need to shut down now. I don't need any more cortisol floating around. So let's shut the system down. So our bodies are really designed to respond to stress, and cortisol helps us deal with stress. So let's say we see a tiger or something scary in our environment, and we need to activate our stress system to run or to deal with the stress. This graph here is showing what our normal stress response looks like, and in the blue is for males, and in the pink it's for females. And basically you see this increase in cortisol once we've dealt with the stressor, and then there's a return to baseline because we don't want our stress system to be activated all the time. So once we've engaged that flight or fight response, we want the cortisol levels to return to normal. So that's what a typical stress response should be looking like. But what happens when you have that toxic stress? When the tiger actually might live in your home, or that environmental stressor is in your home. What happens is that our stress system gets constantly activated. So it impairs that negative feedback loop that's telling the rest of the system, hey, it's time to shut down. And now we're living in this heightened state of stress at all times under these conditions of toxic stress. The second pathway that's important in this framework is called biological embedding during sensitive periods of development. And biological embedding is the process by which these early experiences get under your skin and can alter biology and development. So it's essentially how the early life experiences sort of weave its way into biological structures in such a way that it impacts developmental outcomes. So how does that actually happen? We know that we're born with a set of genes that we've inherited from our parents. There's 25,000 genes that we've inherited from our parents. And that genes are the building blocks, or the segments of all DNA, which contains all information for making proteins. And the number of genes that we're born with are the number of genes that we have for life. Nothing changes in terms of the number of genes that you have or the type of genes that you have. What happens with genes, though, is that they can be switched between active, which means that it directs protein production, or they can be silent. So they can be producing no protein at all. And really the genes interact in very complex ways that are well beyond my knowledge for sure, to perform many cellular functions, and really everything that all our sensory and our language and everything that we do and sort of feel. So the question is how do these genes get switched on to be directing active protein production or off so that they're not? And it's through a process called epigenetics. So this is what I call the Sesame Street version of epigenetics, because I am not an epigeneticist, and I really had to sort of teach myself from a very simple standpoint. So really epigenetics is the change in gene activity that does not change the DNA code itself. So your DNA never actually gets changed, but epigenetics are the chemical changes that influence the way the genes act. So whether or not it's producing proteins or turning it off. So I like to think about epigenetics sort of as a dimmer switch, and that our genes are like light bulbs. So a light bulb or a gene can be totally on, like the light bulb on the far left, or totally off, like the one on the far right, or somewhere in between, which is like that middle light bulb there. So the question becomes like, really what sets that activity of those light bulbs? And it's the dimmer switch, which is the epigenome. So if we think about, that we have these 25,000 genes or light bulbs in our billions of cells, epigenetics are the ones that are constantly kind of turning those genes on and off and dictating how the genes are going to work. Another way that I like to think about epigenetics is that the genome is like the hardware of our computer because it determines the boundaries of what's possible. But we need an operating system to tell the hardware what to do. So we need a program like Microsoft Office or Adobe, to tell the actual hardware what to do. And the epigenome is like the operating system that's directing the function of the gene's DNA hardware. So it's telling the DNA and the genes how to work. Really, the power of epigenetics, it's a really hard concept to kind of wrap one's head around, but it's a really powerful concept when we think about how do like environmental factors influence outcome. So we know that epigenetic marks or markers are the small chemical tags that can instruct the genes to turn on or off and affects the way the cell reads its underlying DNA. So it can prevent DNA from sending messages to other cells. And there are many, many factors that affect epigenetic marks. So things like our diet, exposure to cigarette smoke, using drugs. Exercise is a huge one that can affect epigenetics. Things like financial status, so poverty and psychological states and social interactions. These are all related to the way epigenetic marks can turn the DNA on and off from working. In addition to lifestyle factors, we know that early experiences, either in utero or after birth, can influence epigenetic markers and can lead to, things like preterm birth and birth weight and, our susceptibility to allergic diseases and various kinds of other outcomes. So they have a really powerful influence on people's health and well-being. There's a whole host of articles out there now that really show that early life adversity is associated with changes in DNA methylation, so changes in epigenetics and the way the DNA is working. So studies have shown that maternal nutrition during pregnancy, so a study investigating the Dutch famine at the end of World War II, when women had almost nothing to eat. They looked at offspring at age 60 and found methylation differences that were related to prenatal nutrition, particularly in a gene that regulated insulin metabolism. Other studies have shown that early life socioeconomic status has been related to differential patterns of DNA methylation, so different patterns of epigenetic patterns. And it's not dependent on adult socioeconomic status. It's the early life status that's been related to that. Prenatal smoking, maternal mood during pregnancy have all been related to differences in epigenetic functioning, as well as exposure to childhood maltreatment and institutionalization. So the Romanian orphan studies that have been done in the US, as well as parental stress and conflict, where they looked at maternal stressors during infancy, and they found that there were DNA methylation changes in the children in early adolescence. So one way to look at how early life events might impact later outcomes and epigenetic processes is through DNA methylation, which I just talked about. Another way to look at it is through an epigenetic clock. And epigenetic clocks are sort of a measure of our epigenetic age. And what it does is it captures methylation differences at different sites in our DNA. And clocks can be used as a tool to estimate our biological age, through the patterns of DNA methylation, and that these clocks are then compared to our chronological age. So our chronological age is, our actual age when you say like I'm 38 or I'm 28 or I'm 50. But clocks can actually tell our true biological age. So whether or not we look younger than our chronological age or whether or not we might look older than our chronological age. And these epigenetic clocks can reveal discrepancies between chronological ages and biological ages, and really tell us whether or not someone is aging faster or slower than expected based on their lifestyle and environment and other genetic factors. And rapid methylation aging is associated with increased risk of disease and mortality. So while chronological age is the number of years that a person has lived, so our actual kind of age in years, biological age again reflects the condition of our body and how well it's functioning. So it can reveal again the discrepancy between the two. And there are a lot of different kinds of biological clocks that are out there that measure different kinds of biological aging. One of which is called the PED-B clock, which was specifically designed for pediatric populations using buccal samples, so cheeks samples, to look at developmental biological aging, because a lot of the clocks that are out there are for older individuals. But Michael Kobor, who's a collaborator on this project, looked at developing a developmental clock that would be appropriate for much younger samples, and that's what we used in this study. And again, accelerated biological aging early in life really can influence developmental processes based on environmental factors. So why would some people be older than their chronological age? So why would their genetic clocks or their biological age be older? There are several reasons for epigenetic aging. A lot of them that have been looked at are related to lifestyle and nutrition, and are aspects that can make either people younger or older. So if you're eating healthy food and you're exercising regularly, you can see a decrease in your age. So biologically, you might look younger than your chronological age. If you're not eating very healthy, or if you have a high BMI and have various stressors in your life, then you would see an age acceleration, where you would be looking biologically older than your chronological age. So we actually conducted one study. My colleague Divya Joshi and I used the Canadian Longitudinal Study of Aging, which is a very large sample here in Canada of just almost 1,500 participants who were aged 45 to 85. And they provided blood samples to look at DNA methylation. And we looked at, we calculated two different kinds of clock. We calculated a Grimm age clock and a pheno age clock. And we looked at whether or not self-reported adverse childhood experiences were related to age acceleration. So whether or not they looked, people who experienced higher adversity would look biologically older than their chronological age. And what we found is that the cumulative adverse childhood experiences score was associated with higher Grimm age acceleration after adjusting for covariates like lifestyle factors and socioeconomic factors, so that the more adversity that a participant had reported experiencing in childhood, they had an older looking clock and were older biologically compared to individuals who reported fewer or no adverse childhood experiences. And what was interesting about this is that we found a difference with this one GRIM age clock, but not with the pheno age clock. And we know that GRIM age is associated with risk of mortality. So it's really important to know about biological age and ways to intervene to prevent morbidity and mortality later in life. So just to sort of sum up, like, why is it important to look at DNA methylation even in children? It offers us an opportunity to compare the biological impact of different types of environmental exposures, like physical exposures or social exposures, across development, and to understand their potential implications for health and behavioral risk and resilience. And in the context of an intervention, it provides us with the opportunity to see if we can actually reverse biological impacts that we know are associated with early life adversity. So this boils down to why we did, I know it's a lot of information and a lot of biology and a lot of, getting down to the nitty gritty of the way our cells work, but it really formed the framework of why we would even look at biomarkers within the nurse-family partnership. And our question basically was, can early interventions reverse biological embedding? Because all the data that I had showed you so far were done in cohorts. So they were all associations. So we saw that reports of early life adversity was associated or related to increased risk of something, but there are very few studies that actually show whether interventions can actually impact on reversing some of these biological difficulties. So if we take a public health approach to preventing adverse childhood experiences like child maltreatment, there are various points at which along the pathway one can intervene. And we know that the nurse-family partnership is a primary preventative intervention because it's really targeting prevention before any occurrence of risk or exposure to abuse or maltreatment. It's also targeted because of the population that the nurse-family partnership is offered to. So now I'm going to talk just very briefly about the BC Healthy Connections project, which again was the randomized controlled trial that was done in British Columbia here in Canada and led by Nicole Catherine and Charlotte Waddell. This is the protocol paper that Nicole was first author on and that the team published about the actual trial and the eligibility criteria. So the BCHCP was really focusing on the effectiveness of the nurse-family partnership compared to existing services in British Columbia. And it had several eligibility criteria. So participants needed to be aged 24 years or younger. They had to be a first-time parent, less than 28 weeks gestation at the time of enrollment, able to provide informed consent in English, and experiencing socioeconomic disadvantage if they were between the ages of 20 or 24. So anyone ages 19 or younger were automatically enrolled in the trial, but individuals between 20 and 24 also needed to be experiencing some sort of socioeconomic disadvantage. And we define that as receiving some sort of medical assistance or financial assistance, reporting that they found it difficult to live on the total household income with respect to food or rent, or homelessness, or having unstable or living, like not having a permanent address and having unstable living conditions. The larger trial enrolled a total of 738 mothers and their children, and it was conducted across all 5 regional health authorities in the province. There were 2 adjunct studies as part of the BCHCP. One was led by Dr. Susan Jack, and it was a process evaluation that ran in parallel to the trial itself. And then there was the adjunct study, the Healthy Foundation study, where we were looking at biomarkers in a subset of the participants that had come from the BCHCP. So this was sort of, putting together that framework into our model of the way we were thinking about how, the nurse-family partnership may influence or reverse, I should say, and mitigate some of the risk associated with biological embedding and early life adversity. We assessed a number of adversities. Then there was the randomized control trial. We took a number of different biomarkers, and then the trial also looked at different outcomes at child aged 24 months. So just briefly, for those of you that don't know what we mean by randomized control trial, pregnant girls and women, young women, were invited to participate in the evaluation. If they consented to participate, we took a baseline measure right after they consented. And after that data was collected, then they were randomized to one of 2 arms. And randomization is essentially like rolling a dice, but it's like a one-to-one dice. So they were either randomized into the treatment group, which was the nurse-family partnership, or they were randomized into the control group, which was essentially services as usual that were offered in British Columbia. So that's how we had the 2 groups, and people were just randomly assigned to one of the 2 groups on a one-to-one basis. These were the measures that were taken in both the BCHCP and the Healthy Foundation study. I'm not going to go into a lot of detail. There was, quite a comprehensive assessment that was done on the mothers across time. So prenatally, measures were taken at baseline and at 2 months. At baseline, and then measures were taken at 2 months postpartum, 10, 18, and 24 months postpartum. And you can see that a lot of the measures were repeated across time and were self-report. Child measures were mostly taken at child age 2, and this involved both self-report as well as observational data, where they looked at cognitive development in the infants, and where I'm not going to be reporting on that today because that's part of the larger trial. And then what we did was we took the biological measures, and we took different kinds of biological measures. So we took, to measure chronic stress in the mothers, we took hair. To measure stress in the infants, we took saliva. And to measure DNA methylation or epigenetic changes, we took buccal swabs. So I'll talk a little bit more about that. So why would we collect hair as a measure of chronic stress? Stress actually, cortisol, when it's released in our bloodstream, actually gets absorbed into the hair shaft. So we can actually measure the amount of stress that someone experienced within a 3-month window. So we cut the hair from the back of the head, the posterior vertex of the head, and it's like the size of half of a pencil width that we need to take, and then we essentially grind the hair up and put it through an assay so that we can measure the actual level of cortisol that a mother experienced within a 3-month window or period. We took saliva samples from the infants. As we know, infants produce a ton of saliva, so this was relatively straightforward and easy to do when they were little. It becomes a little more challenging as they get older. And basically, it's like this little kind of cotton swab that they get to suck on. And again, they love it when they're little, not so much when they get to be like 18 and 24 months. And then we put the saliva in a tube, and again, we assay to look at cortisol in the saliva, which is a much smaller measure. It's only within like the last 20 minutes and not 3 months when we take it from saliva. But because infants don't have a ton of hair, this is what we had decided to do. This is our sort of timeline again for the measures and the development. So again, women were recruited during pregnancy. We took the baseline visit. They were randomized into the nurse-family partnership or the services as usual. We took hair samples at baseline, so during pregnancy, and then again at 2 months postpartum. And then we took saliva from the infants at 2, 10, and 18 months. And then we took the buccal swabs, which is like a little paper toothbrush, where we scrape the inside of their cheeks at 2 months and again at 24 months, because we wanted to look both at the sort of potential prenatal impact of the nurse-family partnership, and then at 24 months to look at almost like the full dosage prenatal and postnatal impact of the nurse-family partnership on epigenetic functioning in the kids. This is some data from the larger trial that was reported by Nicole Catherine in an early paper looking at baseline characteristics of the participants in the sample. And what we can see is that adversity was common in this sample and was compounded. So that 70% of the girls and young women were experiencing 4 or more indicators of adversity. So 52 reported less than high school, not surprising given their age. 84% were living on low income, so less than $20,000 a year. 32% reported moderate to severe psychological distress. They were highly, highly mobile. About half of the sample reported moving 3 or more times in the past year. 49% reported intimate partner violence in the past year. 56% reported moderate to severe abuse and neglect before the age of 16 years. So again, a really strong and resilient sample in many ways, but adversity was quite common in this group. So now getting to the results. So we, again, took hair samples at baseline. So prior to randomization, prior to any participant receiving the nurse-family partnership, that's this baseline where we have the pregnant figure here. So we took hair cortisol samples at that point. And here is, the blue line is the nurse-family partnership group, and the red line is the control group. So what we can see here at baseline, there was no difference between the two groups in terms of hair cortisol levels, which is really a good check for randomization, because we don't want to see a difference between the two groups before they're actually randomized into the nurse-family partnership or services as usual. But what we did find is that there was a significant difference between the two groups at 2 months in terms of the hair cortisol levels. And again, this is a measure of chronic stress over the past 3 months. And what we saw is that hair cortisol levels were higher in the control group than they were in the nurse-family partnership group at 2 months postpartum. So why is this important? Because when we look at normal cortisol levels over time for pregnant persons, we see that there's this huge peak in cortisol right at birth, which is this 0 with the dotted line here. And that's because we need cortisol to help with delivery, essentially. But then cortisol levels should actually drop over time, so that over the course of, this is in weeks, so at 8 weeks, which is equivalent to our 2-month sample, cortisol levels should actually be going down. That's the normal physiological response. And what we saw was that in the control group, those stress level hormones were not going down like they were supposed to be. They were actually staying stable and high. So if I go back to this slide here, Again, you can see that the control group pretty much stayed even across time when they should have been coming down. So we saw the natural drop in stress levels in the NFP group that we should have been seeing. Before I present the child outcome data, I just want to make a slight distinction and talk a little bit about intent to treat versus dosage models, because we looked at, we did the analyses in different ways to kind of look at our findings. So in intent to treat models, when a participant is randomized, they are then analyzed according to the group that they were assigned, even if that participant did not receive any of the intervention at all. And the reason that's done, it's the gold standard for randomized control trials. It's so that we're not introducing bias, so that there's nothing that's different perhaps about those participants that did not take up the program or that dropped out of the study, and that it's not due to certain characteristics. So really, intent to treat analysis is the gold standard of analyzing data within a randomized control trial. However, it does dilute the potential power to detect any kind of clinical or statistical differences, especially if the dosage is different within an intervention or treatment, or as dropouts increase across time. So I just want to mention here that retention in the trial was really, really high in the research study. It was, I want to say, close to 90%. But we know that the dosage of the number of NFP sessions that mothers received were different between groups. So that some mothers who were randomized into the intervention arm didn't have any sessions, so they were more like the control group, or might have only had 1 to 2, or it varied across time depending on, prenatal or postnatal, because we know that, the nurse-family partnership is offered across almost 2.5 years, so dosage is going to differ. So we decided that we would also look at dosage effects. So we looked at intent to treat analysis as well as dosage effects. And how we looked at dosage was just within the Healthy Foundation study, within the intervention arm. So for all participants that were randomized to the nurse-family partnership, we looked at the number of sessions across all 3 phases of the nurse-family partnership. So we looked at how many sessions did they have prenatally, plus, during infancy, as well as toddlerhood. And then we looked at, we did a statistical analysis, which sort of showed 3 different groups, essentially, within the Healthy Foundation study. So one was a relatively low dose group, so almost 33%, it's this red line here, had less than 10 visits prenatally and in infancy, and dropped off essentially during toddlerhood. So that's our low dose group. Then we had a moderate group, which is this green line here, which represented 56% of the participants who had above 10 visits during pregnancy. They had a bit of an increase during visits during infancy, and then it stayed relatively the same number of sessions in toddlerhood. And then our final group here was what we called our high dose group, which is this blue line, and it's only 11% of the participants, where they had 16 to 20 prenatal visits, and then they jumped up quite high in infancy, where they had a number of, close to 50 visits over the course of infancy, and then a bit of a decrease again during toddlerhood, but it was still pretty high because it was more than 20 visits. So that was our high dose group. And we decided to analyze our child data in two ways. One was the intent to treat model, where we looked at the nurse-family partnership versus control that was only based on randomization status. And then we also used this dosage analysis. But in this case, we collapsed control and no visits, and low and then moderate to high were grouped together. So those are the results I'm going to compare now. In terms of the changes in the infant saliva sample over time, so time is represented here. Again, we took saliva samples at 2 months and at 10 months and at 18 months. This data only represents what we call basal saliva sample, because what we did at each of the visits was when we went into the home, we took a saliva sample from the infants, then we exposed them to a little stressor, and then we took saliva samples at 20 and 40 minutes post-stressor. But when we looked at the data, there was a lot of missing data. And I think in part it was because we were doing these visits in the home. In part it's because, especially at 10 and 18 months, the kids don't necessarily like us taking saliva samples from them. And so it really invalidated the data when we had so much missing data. So we only looked at the basal level of cortisols over time. And here we looked at intervention status, where we just looked again at the nurse-family partnership, which is the blue line here, versus control. And what we can see is that at 2 months, the infants that were in the nurse-family partnership group had slightly higher levels of cortisol compared to the infants that were in the control group, again, only at 2 months, but that this effect sort of dissipated and disappeared over time, so that by 18 months, we didn't see any difference between the two groups. If we look at the dosage analysis, where again, we're looking at changes in infant saliva samples of cortisol over time, but this time we're looking at dosage. Here we can see that, you know, the control group, or the ones that had no NFP visits at all, again, had the lowest level of basal cortisol at 2 months, and that the low group kind of fell in between the control, and that the moderate to high number of sessions group had the highest basal cortisol at 2 months. But again, that these effects sort of kind of decrease and disappear over time, although they're still maintained a little bit more here when we look at the dosage analysis at 18 months. In terms of the epigenetic clock, we did not find any. Here again, we're looking at whether the difference between biological age and chronological age. And we found no difference between the NFP versus control group if we did the intent to treat analysis. But if we looked, and again, we took samples at 2 months and at 24 months. If we look only at 24 months and we're doing the dosage analysis, what we're actually finding is that infants in the control group, or who had no NFP visits, look biologically older than the children who received, who were in the moderate to high number of visits group, and that again, this low kind of dosage group kind of falls in between. So we're finding this really quite a beautiful dosage effect here, and that again, the ones who were in the control group look biologically the oldest. Okay, so why does maternal stress matter, and how might this be related to long-term outcomes? We know that from other work that maternal distress, whether it's measured by adverse life events or early life adversity, is associated with higher stress in mothers, which then might be related to nutrition intake, sleep disruptions, reduced physical activity, and that this is all related to fetal development, so that fetuses who are exposed to high maternal distress in utero have lower heart rate variability. There's lower coupling between maternal and fetal heart rates and higher heart rate reactivity, which is then associated with difficulties in birth outcomes, so lower birth weights, and in terms of postnatal exposure, there's an increased risk as well in terms of maternal and infant interactions, because we know that women who are experiencing high levels of stress and depression have a higher risk of having attachment and bonding difficulties with their children, which then might be associated with a number of child outcomes. So again, we want to be intervening as early as possible in order to be disrupting this kind of pathway of risk for children. So when we think about, the hair cortisol data from our study, we saw that the nurse-family partnership made a difference in terms of reducing physiological stress in mothers. This is another diagram from a review article by Charles Nelson, again showing that framework about how early life adversity can be related to epigenetic changes in our stress system functioning and immune system functioning, and then how those changes are associated not only with developmental outcomes, but with adult outcomes later on. So again, we want to be intervening at this point. When we looked at the dosage data, we saw that, children that were in the control group were biologically slightly older than the high to moderate dosage group. So we want to be intervening as early as possible to prevent this kind of epigenetic changes and changes in the underlying biological system from happening. So what's potentially going on? I would say that the saliva data from the infants has given us a bit of the biggest challenge in terms of trying to interpret it, and the literature doesn't really help us figure it out. The literature itself is quite messy and doesn't give us clear answers. We have looked very closely at other intervention studies that looked at HPA axis functioning and cortisol in infants. And there's basically consensus that there's remarkable inconsistency with respect to how interventions influence cortisol. Some studies find that interventions do have an impact and other studies don't. One meta-analysis, which is a synthesis of a number of studies, didn't find any positive effects. Again, our stressors were probably not powerful enough to create a stress response in the infants, but again, we had a lot of missing data so it was very difficult for us to interpret. We also know that this was a fairly, again, high-risk sample in terms of adversity, even at baseline. So maybe the risk is almost too high and that an inclusion of a low-risk comparison group might be helpful. When we looked not within the intervention data, but at other data, we know that those low baseline levels are associated with risk of negative developmental outcomes in children. So there's a very large study that's has been done in the US where they found that low baseline levels at 7 and 15 months were associated with lower cognitive scores and higher behavioral problems and disorganized attachment in infants. So again, we saw the lower baseline at 2 months in the control group infants, which changed over time. When we looked at dosage, we saw the lower levels were at 2 and at 18 months in the control groups or who did not receive the NFP, but higher. So again, it might be related to developmental outcomes over time. Why does epigenetic change matter? Again, we know that prenatal stress and psychosocial stress during pregnancy can alter epigenetic and DNA methylation in the infants, both in terms of epigenetic genome-wide studies as well as DNA methylation studies that have looked specifically at candidate genes, which is not what we did. We looked at the clock right now. And really, it's a question of how does this impact child health outcomes over time? How is it related to stress reactivity and later internalizing and externalizing problems and cognitive functioning? This is a sort of adaptation on Heckman's equation. Heckman is an economist who really makes the case. I know I'm speaking to the choir here, but that it's really, really important to intervene early. I mean, we are seeing changes very early on in development and the potential of the NFP to be impacting, those biological markers. So I think it adds to the case of why it's so important to intervene as early as possible. Because as we can see, the earlier you intervene, the lower the risk is over the life course. If you wait until childhood or adolescence or even later in adulthood, risk increases and it becomes more and more difficult to make changes. Again, not impossible, just more difficult. So in terms of next steps and where we're thinking of analyses, but I'm again really hoping that this can be a conversation and you guys can give us direction about what you found interesting or what you would like to see in the future. We're hoping to look at these outcomes in the context of changing adversity over time. Again, the BCHCP collected a wealth of data at multiple time points, so we can look at, does adversity change over time and does that impact child biomarkers over time? And we're also going to look into conducting analyses where we're looking at whether or not those biomarkers are related to the 24-month child outcomes, the cognitive and internalizing and externalizing outcomes at 24 months. So that's sort of where we're looking to in the future. Some of the questions that our team has been thinking about since we've analyzed the data is, how do we potentially prevent the relapse of risks postnatally? How can we continue to engage mothers and promote resilience? What extra resources might be needed for young mothers who have experienced their own early life adversity? And, we recognize the limitations of our study markers. It really captures only a small snapshot in time. But, it's a different angle than self-reported stress and distress factors. So what unique variants might we be capturing with our physiological measures of stress? So I'm just going to end off by really making a case about why I think that inclusion of biomarkers is important, really because it provides a scientific explanation for specific types of health outcomes. It provides the basis for assessing individual susceptibility to early adversity. It provides, and I think this is one of the strongest cases, is it provides additional and objective indicators for assessing intervention effects. A lot of the interventions that we evaluate within a psychosocial context are typically based on self-report, and this is a whole other angle that's really objective. It can increase the leverage to advocate for preventative interventions. So if we're making a case that the NFP can reduce stress, in new mothers, we now have some physiological evidence that suggests that. And it provides early evidence of effectiveness in reducing disease risk, which we might not see for many, many, many years, which David's original trials have certainly supported, but now we're seeing maybe the early biomarkers of those findings. So thank you. I'll stop there.
Right. Andrea, thank you. Thank you so much. I mean, thank you for not only selecting this forum as the first place to share these novel and exciting findings, but thank you for the masterclass on epigenetics and really your description of the stress response and the HPA axis. I've been a nurse for over 30 years. That is the clearest explanation I have ever heard that has really resonated. So thank you for so skillfully teaching us and sharing these findings. And before we open it up for a few minutes of questions to you, I'd like to ask David if you could share some of your reflections or remarks as you hear these findings presented for the first time, and to place them into context for the broader NFP global community.
Well, Sue, and thank you for that. And Andrea, thank you so much for this wonderful presentation. I'm with Sue. This is clear and very useful. My first set of questions has to do with just the putting these differences in levels of service received on the part of the intervention group in context. In other words, are these people who remained accessible to you for the remained accessible for the nurses to serve them, or was this a function of moving out of the area? That's my kind of first question, because I think it has a bearing on how one goes about interpreting this. And the other question is kind of in a way parallel, and that is to what extent um, was the 90% retention rate, did it apply to gathering the biomarker data? In other words, did you see higher rates of non-compliance, or people who didn't want to provide the hair samples or the buccal swabs for the infants? Were the rates still 90%? And if so, how did they differ? So these are I'm wondering more generally, I suppose, about the role of selection bias affecting this pattern of results that you see here. And I don't want to take up a lot of time, because this is really important, and I don't want to interfere with other people's comments. But this is those who know me, these are the kinds of questions I typically ask. So thank you.
Yeah, no, thank you for those questions, David. So I'll try to answer them, but I can say that if a participant moved outside of the health authority or the catchment area, we did not collect data on them anymore. I think that they could be transferred to a different nurse if they were, but if they were outside of the catchment area, we weren't collecting data. And I should have mentioned that even though the NFP was offered and the BCHCP was offered across all 5 health authorities, the Healthy Foundation study data only came from 2 of the health authorities, Fraser Health and Vancouver Coastal, which is where the majority of the participants came from. And that was because we had, that was the opportunity that we had to do the home visits, and we needed to be in the home in order to collect the data. And that, we still got a good proportion of the original sample, the 736 in the BCHCP. In the Healthy Foundation study, we had 385. So the majority of women who were offered to be participating in the adjunct study actually agreed to participate in the adjunct study. So in the 90% retention rate is related to research only. When we talk about the dosage data, we got that from the home visit encounter forms and the alternate home visit encounter forms. And that's what I was calling a session. And that's how we analyzed that data. In terms of higher rates of non-compliance, when we launched the study back in 2016 or 17, I think there was real concern from the research team that mothers would, be unwilling to provide their hair especially. A lot of moms are willing to let us take saliva samples or cheek swabs from the kids, but their hair in particular. But we had a very high rate, not only because we had an opt-out option. So even if they agreed to participate in the Healthy Foundation study, they didn't necessarily need to participate in every aspect of it. They could have opted out of hair, but we had a very high rate of mothers, giving us their hair. And the missing data with the infant saliva samples is more like a methodology kind of issue where they might have eaten or been sleeping or needed to feed, or we didn't have enough saliva, or something went wrong in the home visit during that. So I don't think it's a non-compliance issue. I think those are all very good questions, but yeah, I don't know if that answers your question, or Divya, or I don't even know if Nicole's on here, if you have anything to add.
I'm just thinking that those are that line of inquiry from where I sit would be helpful to really be able to thoroughly interpret these findings. And so if it's
Yeah.
I'd love to hear more about this, and I apologize for opening up with questions that are going to be so time consuming. But it's something that we should dig into. I'd love to hear more about where this goes.
Yeah, no, thank you, David. And really, you've even this is really helpful, because when I quoted the 90% retention rate, that's from the larger trial, and we have to probably double check that for the Healthy Foundation study as well.
And the retention for just one last kind of relate is, were they equally retained for these biomarker assays in the intervention and control group?
I believe they were. And I see Divya's nodding, so I'm going to take that as a yes.
Okay.
She's my
All right.
go-to.
I'll shut up and let other people ask their really important questions here.
Okay, thank you.
Great, thank you. Now, Andrea, I'm going to there's been one question that so far that's been posted in the chat. And if anyone else has questions, please either post it in the chat or use your raise hand icon, and I'll call on you. And the first question, again, takes us back to your masterclass in epigenetics. But the question is, after an epigenetic gene is turned on through adversity, can it be turned back off?
So I don't think, I tried to break it down into the most simple way possible, but I think that it does work. And I don't know if I mentioned this, like if we think about the dimmer switches and the light bulbs and stuff, it works like a master control thing. Like it's not, and I think we don't have enough knowledge about how that whole switchboard works well. But, and I'm looking at Divya here, my sense is once the methylation changes have been made, and I don't know that how many people have looked at methylation changes like longitudinally or over time to see that effect of how the patterns might change over time. But once, it's changed, I think it's generally hard to change. And because I remember having this conversation a long time ago with Michael Meany, who brought this really to my world through his rat research. And because I was very interested in this whole idea of reversing, early life adversity. And it was really through drugs or, some, I think, through exercise. But yeah, that's a great question, Divya. I don't know if you have anything to add.
Yeah, no, I think everything in the literature suggests it's very hard to change, but any changes would have to be through lifestyle and environment modifications or diet, exercise, and would have to occur quite early.
Great. Thanks, Divya. Thanks, Andrea. Andrea, if you're in the chat, you'll see the next question right at the bottom of the chat, and I'll read it to you if you're scrolling down. But this is a question from Cynthia, who's wondering if protective factors are measured in the study, and what inherent strengths do families bring in addition to their risks that have been created by our global systems?
Yeah, so thank you for that question. So there are protective factors that were measured in the BCHCP, and it's something that we have to look at a bit more thoughtfully. If people have any ideas of what you think protective factors are, please put them in the chat and we can definitely look at it. Two that we have looked at just from a baseline perspective was self-mastery. David, I'm looking at you now because it was the measures that you took where you were looking at not dosage effects, but women who benefited more from the NFP. It was self-mastery and self-efficacy, I think. Oh, you're on mute, sorry.
Yes, self-efficacy and mastery are two interrelated concepts. The other is intellectual functioning and mental health.
With the Shipley, yeah.
Yeah.
So we've looked at that, but only at baseline, and we were sort of just looking at it in the context of the mother's self-reported experiences of childhood maltreatment in relation to the baseline measure of hair cortisol, and we did not find any protective effects. What we have not done is looked at, how the NFP might interact with other protective effects to have an impact on biomarkers over time. And I think that is the next step for us. So.
Great, thanks. Great comment in the chat from Justine Menzies, and really acknowledging and validating the importance, some of the limitations of short-term evaluations, of early years evaluations that have mixed results, but really validating the importance of, doing ongoing later evaluations to uncover, impacts on children's physical health and education, and then has provided a link to some longer term evaluations with Sure Start, not NFP, and thanks for acknowledging that. And Andrea, can you speak as well to any, the importance of, or opportunities to look at the sample long term?
So I believe that the BCHCP team will be following the sample long term. I don't know if that's with administrative data similar to what they have done in the UK. We are not planning to follow up with any biomarker data, unfortunately. It blows my mind when I think that the kids are already 8.
Wow. Yeah. No.
It's a lost opportunity, but 7, 6, 7, 8, but still, yeah.
Yeah. And I mean, just understanding how you collected the data and the analysis. I mean, it's a complex data set, right? And the challenges of following the families over the long term.
Yeah, absolutely. And I think, had we had we had the opportunity, we could have introduced different types of biomarkers too. And one thing, a limitation from the study is we didn't collect genetic data, which we should have looked at in the context of epigenetic data, but we didn't do that. So, yeah.
And I have a question for you. When I quickly scanned to see who was on the call, and so excited to see such a diverse range of both clinicians, nurses, policymakers, as well as researchers. I did note that there's many professional practice leads, nurse managers, and nurses today on the webinar. If you were to give them 2 or 3 key statements that they could take back to their teams about these findings and NFP, what would be those really clear statements that they could take back to their teams about the findings?
Yeah, I think for me, one of the striking findings, and it was an early finding, was that hair cortisol data, which, we've taken hair cortisol in a lot of different studies, including a Brazilian sample that has 2,000, I think, mothers and children, and we've had a hard time finding effects. So to find, this difference in the NFP, it's a small difference because it's hard to say what, that little bit of lower cortisol means, but that it's actually reducing stress in the moms. And we don't, again, because we took it at baseline and then at 2 months, we don't know when that stressor might have happened. But if we're reducing stress during pregnancy, then that means that we're changing the in utero environment for the child, right? The infant. And then that has huge implications for biological development and outcomes later in life. So I think one of the key things for me was if the NFP was, managed to move that marker a bit, that's quite striking. And I think the other striking, and again, going back to David's point about framing it in, with a cautionary tone, because we looked at dosage and we need to, I think, delve deeper into what dosage means. That we found even a slight difference, again, that the control children were biologically older when we looked at the PED-B clock. I think that was a pretty big finding too, because we know that epigenetic clocks are linked to later morbidity and mortality. So
Great. Thank you for summarizing that, because I think in the work that nurses and managers and supervisors do, they are in the prime position to share with families as well as policymakers up why this program is important and the benefits and the effectiveness and really the robust evidence that underpins it. So anytime they can have some clear statements like that to help explain is I think really valuable to ensuring that this program is available and offered to so many other families.
Well, sorry, I'll just add that one of the things we're hoping to do is once we've vetted this through David and all the other research team members, and it's, gone through peer-reviewed publication, that we'll be creating infographics from that. And I'm happy to share that with this group because it'll hopefully give the bite-sized information and key messages as well.
Oh, that's great. And then just in our last time, there's one last question. Again, it's in the chat if that's easier, right at the bottom for you to look at, Andrea. But it's infants in the intervention group had higher cortisol, and can this be related to selection bias of the sample, or what other factors might explain the high baseline levels?
So I think, that's a fantastic question. And we really went back to the literature because we were trying to make sense of what even just a basal cortisol level meant in this age group. And like I said, the literature is messy and there's just no consensus. So I really hesitate to try to interpret it at all. So because some people might think that higher cortisol levels are negative because you don't want to be operating at a high level. But then we also know that blunted levels, because it's more of a measure of the way the HPA axis is responding to stress over time, it has that dampened effect because that negative feedback loop that I was talking about doesn't get turned off, right? But then we saw that there was that the groups came closer together, by 18 months. So I don't even know how to interpret it. And I actually hesitated in even presenting it because I just don't even know how to make sense of this data. So I'm sorry, I don't think it's a selection bias. I think it's a state of really what does cortisol in this context at this age mean. And I should say that we also we did control for things like time of day of awakening and naps and all of that, so that all those covariates were controlled for that we know affect the HPA axis. But I really can't make an interpretation on it. So.
Well, that's the great thing of research and science is one study creates more questions.
Yeah.
And one more question, just because it's there. If we see that change in the clock at 24 months, it makes me think, what might we see later in the life course? Has there been any other studies that show differences at such a young age?
Not in an intervention context. No.
Okay, great, super.
So, but David has his effects within.
Yeah, I just wanted to say, Andrea, I'm so glad that you presented the whole picture, all of the data, because that kind of thoroughness is what we need, even if it's unclear. It provides us with some an appreciation for the complexity of all of these things. And I just want to thank you again for a wonderful presentation, great interaction with the audience. And it kind of reminds me, I suppose, that in this work, no one has all the answers, and that we are stronger together. And this illustrates that the fact that we have a shared enterprise in some ways, and together we're going to get it right, but it's going to take a little while, I think.
Totally agree. That's an excellent note to end off on.
It is. And thank you for that note to end on. And Andrea, thank you so much for, again, choosing this forum to share these very exciting early results. It's such a pleasure to listen and learn from you. So thank you for presenting today. A huge thank you also to Anna Lindberg, who coordinates the webinar series and ensures that everyone had the registration and that all of our technology worked today. We couldn't do this work with her. And thank you to all of you out there who joined us today to listen and learn and to ask your questions. So greatly appreciated. And stay tuned. Our next webinar will be in March 2025. And if you registered for this one, I imagine you will be getting the information about the next webinar as soon as the date and the speaker is confirmed. So stay tuned for March. And thank you all, and wishing you all a peaceful holiday season. So take care and goodbye.
Thank you. Bye.
Bye.